Hello, my name is Katherine Do, and I work with the Galaxy for Proteomics group at the University of Minnesota. I'd like to welcome everyone to the overview video for the Clinical Metaproteomics tutorial. This tutorial is made up of 5 modules. Following this overview video, we will cover each module in its own video. The agenda for this video includes some background on the metaproteomics field, such as why we developed this workflow, and an overview of the clinical metaproteomics workflow. This will lead us into the hands-on section, which will introduce the GTN website, where we host the tutorials, and the Galaxy EU website, where we will run our analysis. First up is background. Mass spectrometry-based metaproteomics enables the analysis of proteins expressed by microbial communities and can be applied to clinical samples to understand microorganism contributions to disease. Metaproteomics is the large-scale characterization of the entire complement of proteins expressed by microbiota. Clinical metaproteomics provides insight into host microbe dynamics in disease, such as how the microbiome responds to its host environment. However, there are numerous challenges. One example is the use of very large protein sequence databases. The size of these databases are often in the range of millions of sequences, which many tools cannot handle. Moreover, searching these large databases can decrease sensitivity during peptide and protein identification from mass spec data, while increasing the potential for false positives. Another challenge of using clinical samples is that the high relative abundance of human host proteins can hamper the detection and identification of lower abundant microbial proteins. To address these challenges, we have developed a novel clinical metaproteomics workflow that provides customized bioinformatic identification, verification, quantification, and taxonomic and functional annotation. This bioinformatic workflow is implemented in the Galaxy ecosystem and has been used to characterize diverse clinical sample types, such as nasopharyngeal swabs and bronchoalveolar lavage fluid. Here, we will demonstrate its effectiveness via the analysis of residual fluid from cervical swabs. Before we get too far along into the tutorial, I do want to make an important note. To demonstrate this workflow, we will analyze a small example set of quantitative proteomics data from an ongoing study of ovarian cancer in fluid from cervical swabs. This demonstration is meant to highlight the methodology and its effectiveness. It is not meant to communicate biologically significant results or new discoveries. During the tutorial, I will mention details such as how many peptides were identified or organisms identified, but these are meant to inform the audience of where we are in the workflow, what kinds of outputs we are generating, and what we are doing. So, moving on, this tutorial is broken down into 5 modules: database generation, discovery, verification, quantification, and data interpretation, which I will cover on the next slide. This is an overview of the entire clinical metaproteomics tutorial. I will summarize the modules here, but keep in mind that each has its own dedicated video. Starting from the top left corner and going clockwise, the order of the modules is as follows: database generation, discovery, verification, quantification, and data interpretation. In the first module, database generation, we want to obtain a comprehensive protein sequence database, which would include proteins from humans, contaminants, and our various species of interest. In this example, we didn't have a reference database or information from 16S rRNA sequencing data, so we generated our own comprehensive database using a literature survey. If 16S rRNA data is present, the taxon identified can be used to generate a customized database. As the size of this comprehensive database is generally too large, in the range of millions of sequences, we will use the MetaNovo tool to reduce the size of the database. This reduced database will then be used for the discovery module. The discovery module, as you can see in the top right corner, uses two peptide identification programs, SearchGUI Peptide Shaker and MaxQuant, to identify peptide and protein sequences via sequence database searching. This will allow us to successfully retrieve reliable microbial peptides. Next is the verification module in the bottom right corner. In this module, we will use PepQuery 2 to verify confident identification of microbial peptides to avoid misassignment of human peptides. This is a very important quality control step that ensures the trustworthiness of our findings. We will then create a database of protein sequences associated with the PepQuery verified peptides. This verified database will help us reduce false positives when we quantify our proteins and peptides. Then we have our quantification module in the bottom middle. One objective of running a quantification workflow is that we are able to measure and compare the abundance of proteins or peptides in biological samples. Here we will use MaxQuant to quantify microbial and human peptides and proteins separately to compare results. Lastly, we have our data interpretation module in the bottom left corner. In this module, we will perform statistical analysis using MSStats TMT and taxonomic and functional annotation using UniPept. I do want to point out that the Clinical Metaproteomics tutorial includes all 5 modules. I mention this because each module has its own workflow link that we will import. While we can create a giant workflow to contain all of these modules, creating a separate workflow for each module makes it easier to catch errors early on before moving on to the next. So keep in mind that each module has its own workflow, and these workflows together make up the Clinical Metaproteomics tutorial. Next, we will move on to the hands-on section. For this section, I highly recommend you open different tabs to make things easier for yourself. We will cover the basics of the GTN website and Galaxy EU, such as how to navigate to find things, and a preview of how we will set up for each module. That will prepare us for the next video, where we cover Module 1 database generation and execute its workflow. Here I've provided the links for the GTN and Galaxy EU websites. First, let's visit the GTN homepage. As you can see, there are different categories, such as Introduction to Using Galaxy, different fields, technologies, and resources for admins, developers, and instructors. For us, we will follow a learning pathway. You can click on this button in the top bar or on this large icon on the right under Quick Start. These learning pathways are sets of tutorials that are intended to be followed in the order that they are listed. They are meant to give you the basics of certain topics, so they are ideal for beginners. The one that we want is called Clinical Metaproteomics Workflows within Galaxy. Click on it and it will take us to the tutorials. For each module, you can click on its tutorial link. If you have any questions about using Galaxy, there is an FAQ section. You can go back to the top bar, click on Help, and then FAQ, and then Galaxy FAQs. Next, we will move on to the Galaxy EU homepage. This is the Galaxy instance we are using for this learning pathway. You will want to make an account or log in to your existing one. If you are making an account, you will need a valid email address. Now, let's head back to the GTN website and see what a tutorial looks like. I'm showing the first one for database generation, which we will walk through in the next video. At the top, there is the overview box, which has helpful bullet points such as questions we address and the objectives for this tutorial. There are also links to other tutorials that may help beginners learn more about Galaxy. Then, under the overview box and on the left-hand side, you can quickly jump to other sections by clicking on these headers. Throughout this tutorial, there are boxes that include tips, common questions, and solutions to help you along the way. We've also included the parameters for the tools used. Next, we will quickly summarize the setup for each tutorial. For setup, we will use the same one for all modules. Step-by-step instructions will be covered in the next video. First, we will create a new history for each module, for a total of 5 histories. We do this because it's much easier to work with 20 to 30 files rather than hundreds. We will also use separate input datasets and workflow links for each module. So, for each module, you will create its own history, import that module's input data, and run its workflow link. I also want to note the difference between a history and a workflow. I have added a little scroll icon on the left to represent a history. You can think of it as a record of your analysis, similar to a search history on your web browser. This tracks the files you've used and generated, all of the runs you've done, including things that worked and didn't work. In the middle is a folder icon, which represents your data sets. These you will record in your history. And then, on the far right, I have an icon to represent a workflow. A workflow contains a series of tools represented by the shapes that have been preset with parameters, and once you start a workflow, it will run each tool automatically. This wraps up the overview video for the Clinical Metaproteomics tutorial. In our next video, we will cover Module 1, Database Generation. I have included the link here in case you need it. We hope that this overview was interesting and hope to see you in the next video.